Ladies and gentlemen, welcome to my presentation on Jacuzzi 2, which is a software for RNA modification mapping on the Illumina or Nanopore platform. My name is Christoph Dieterich and I'm based in Heidelberg. My lab is excited about RNA research. As probably all of you know, RNA modifications occur on different RNA species, for example, ribosomal RNA, messenger RNAs, and tRNAs. And that is subsumed under the terminology epitranscriptome. These modifications are brought about by so-called modification enzymes that implant in a site-specific manner modifications on the respective molecules. So that may affect different aspects of, for example, messenger RNA life, such as coding, sequence, half-life, turnover of messenger RNAs, also splicing or the translation of messenger RNAs. For ribosomal RNAs, that may affect ribosomal assembly and ligand interactions. And lastly, for tRNAs, we see an impact on folding, amino acylation, and translation, as has been reported in literature before. Now today we would like to focus on messenger RNAs, and we are interested in identifying modified residues on the respective molecules. Here you can see 4 different types of modifications, and the big question is obviously what are relevant and well-working approaches to identify and find these modifications. So today I would like to focus on N6-methyl adenosine, or in short M6a, as a modification that is more abundant than any of the other shown here. So of course there have been several methods proposed for profiling the epitranscriptome, and a whole series of methods is based on Illumina sequencing where so-called reverse transcription signatures are interrogated. That could either be in an antibody-dependent way, as for example with the myClip approach, when M6a recognized an antibody is used and cDNA retruncations are monitored as an indicative signature, or you have a differential digestion assay where an enzyme called MADS-F recognizes methylated versus unmethylated RNA and differentially cleaves this RNA. Or also another possibility would be DART-seq, which is a transgenic approach routinely applied in cell culture, where basically a reader protein is fused to a base editor of a different kind, and that signal is then visible in the cDNA library on the Illumina sequencer. And of course there are many more approaches that you can find in literature. Now, as an alternative, a very recent alternative, I should say, Nanopore direct RNA sequencing was introduced, where a single RNA molecule is threaded through a nanopore. And basically, since there's no PCR or reverse transcription involved, RNA modifications are preserved on this molecule. So typically, the pore extends over 5 consecutive residues on the RNA molecule, meaning any changes that occur within this 5 mers while the molecule is passing through the pore can be measured by the device and then can be turned into base calls by the company's software, for example. So we entered the field of RNA modifications with the software Jacuzzi at the time, back in 2017, where we focused on RNA editing as principal event, so either an A to I transition through a deamination or a C to U transition. And those events are visible in cDNA libraries again through base mismatches. At the time we were comparing essentially the frequency of mismatches with a statistically principled approach using the Dirichlet multinomial distribution, either across conditions or in a comparison of RNA to DNA sequencing. So we now moved on to Jacuzzi 2, which is basically our new kit here on the block, where we extended existing program modes, so-called call1, call2 modes, by adding mismatch rate comparisons or insertion rate comparisons and also by entering entirely new program modes like read and coverage profiling, or so-called RT-arrest running modes. So here essentially we look at piles of reads again in a comparison across two conditions, and we are asking basically whether any coverage drop that we observe here at a given particular position is significantly higher than you would expect by chance, using again a principled statistical modeling approach called the beta binomial distribution as underlying modeling framework. Now the last addition is a so-called base stratification running mode. Here essentially we can already tag and separate reads from read alignments based on the existing pre-existence of a given substitution, for example, an A to G substitution. And that could be used, for example, for metabolic labeling experiments where characteristic changes are brought about, and they could be then contrasted over two conditions, whether they are changing dependent on the modification status and whether we are looking at a newly synthesized RNA, for example. So in principle, we conducted a major overhaul of Jacuzzi. We added many more features, and we are now looking at a software package which is much faster than any of the previous versions. We are quite flexible, that we allow for mixing of different library types, and we keep our statistical modeling approach in a very principled way. So using either the directly multinomial distribution for base substitutions and the beta binomial distributions for coverage changes or insertion deletion events. This has been complemented now by an R package, which we call Jacuzzi 2 helper package, which facilitates easy downstream analysis of the software output of Jacuzzi 2. And let me just focus for a bit on speed as one of the relevant factors. So here you see in red the original Jacuzzi 1 version during the time of publication. Then in green, improvements that were gradually introduced over time, and in blue you see the latest version. And as you can see, in the era of multi-threading, it finally makes sense to use several CPU cores with our latest version, so processing speed, regardless of the library type, becomes considerably faster. Now let's move back to our initial question, which is M6a detection. So here basically I would like to introduce to you differential digestion assay on RNA using the MATS-F enzyme, and also in combination with the demethylase enzyme FTO. So the signature that we hope to cover here is a coverage drop. So how is that affected? Basically on the left-hand side you see M6a highlighted in red on messenger RNAs. Then you split your sample, you treat one half with FTO and the other one you mock treat. And then both of these parts undergo a digestion with MATS-F. And the result of that is turned into a next generation sequencing library. Now MATS-F essentially recognizes at a given ACA motif whether the initial first A here is methylated or not. It preferentially cleaves these motifs if the A is not methylated, and it doesn't cleave these sites as well if the A is methylated, so if you have an M6a here. The other approach that we are looking at today is start RNA sequencing, and that is based on a fusion protein, which is basically a transgenic approach where we introduce into a cell of interest YTH reader domain that recognizes M6As fused to a base editor, which is in this case here an ApoBec1 protein that turns a C into a U, which is nearby. So if you turn this approach into a next generation sequencing library again, that would mean that if the reader protein recognizes an M6a site, a nearby C to U editing event should be detectable. Okay, So how do the results look like actually? So for the MUDS-F digestion analysis, you can clearly see on the left-hand side where the FTO treatment removed M6a, a higher arrest rate, meaning more cleavage in these respective 3 replicate samples over the control 3 replicate samples on the right-hand side. And that increase is clearly dependent on the recognition side of the enzyme, here shown in blue. On the right-hand side, the same analysis in Jacuzzi was done with DART-seq data, where you have an active YTH binding domain and where you have a mutated YTH binding domain. So here again, you can see at the characteristic C to T transitions, and if you deviate from the diagonal to the top left corner here, you have an elevated C to U transition in the active YTH binding case, and that would be what we consider positives. All right, so if we integrate the data from both of these experiments, we see a considerable and significant overlap. But we also see that the DART-seq approach yields way more data in a sense that it predicts more target sites and target genes. And the MUDS-F approach seems to be a little bit more conservative, but that might be also because the recognition side is limiting the search space here. If you look at the closest ApoBac editing event to any given significant MUDS-F cut site, we don't see any major difference whether the motif is present or absent. But more than 50% of the sites show this co-occurring event in less than 200 base pairs spaced apart. Now let's move on to the Nanopore. I was already introducing to you this technology where a single molecule with base modifications preserved are threaded through a nanopore. And here the approach is, again, a comparative approach where we're using HEC293 cells again, the wild type situation and the knockout situation, and the M6a discovery is facilitated by looking at characteristic read signatures, which are either insertions, deletions, or transitions. As I told you previously, a 5-mer is recognized by the nanopore, so we are using some contextual information with a central A here, and we're using non-negative matrix factorization to decompose the signal, which is an error profile, and we decompose it into an amplitude and a pattern matrix, which we're actually interested in. So a prominent pattern that emerges from this NMF approach is shown here as pattern 2, where at the central position you would have an A, and deletion rate and substitution rate seems to be increased, and this is consistent across 2 independent experiments. And this particular pattern is also strongly enriched for so-called Drach sites. Drach sites has been previously described as potential recognition motif of the methylase on the RNA. So how does it lead to prediction of new sites? So we were using basically pre-existing clip data from the same cell type, and the score distribution of pattern 1 is shown here as a cumulative score distribution in blue. So all of the other sites that have not been reported by the clip approach are shown in gray, and you can easily appreciate that although fewer in relation, but there are still several high-scoring sites existing in this distribution. And I would like to give you one example here, which is essentially the Ef2 Gene Locus, where on the left-hand side you have a previously characterized M6a site. In blue here, this is the clip signal, and basically you have the wild-type situation on the top panel and the knockout situation in the lower panel. And I think you can easily see that there are a lot of changes in terms of error distributions here. On the right-hand side, you see a newly identified site with a Drach motif, which hasn't been characterized before and which happens to sit in the same gene locus. With that, I would like to close my presentation, and I hope I could convince you that Jacuzzi is a versatile software solution and comprehensive analysis framework for RNA modification detection, no matter whether you're using Illumina-based approaches or Nanopore-based approaches. Software is freely available through this GitHub link here, and the helper package for downstream processing can be found at the same place, basically. Let me close my presentation with thanking the people involved, Isabel Nahmann de Vries, Michael Pichotta, Chi Wang. I would also like to acknowledge funding from the funding agencies, and I would like to thank you for your attention.